You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast, and I have Lowry Curley, CEO of AxoSim, and the website is AxoSim.com. So Lowry, thanks for coming. How are you doing today?
I'm doing great. Thank you very much for the time.
Yeah, so tell me, what's the premise of AxoSim? What do you guys do?
Yeah, the premise of AxoSim, it really starts with the fact that animal models are terrible predictors of how new drugs will work in humans as they're going through the research and development process. And unfortunately, for neurodegenerative diseases like ALS, multiple sclerosis, Alzheimer's, this failure rate is incredibly high. 94% of the time, a drug that goes through all the testing in the lab and then ultimately goes through animal testing, 94% of the time it doesn't work in humans. The fact is animal biology simply isn't human biology, and that's really the problem that we've set out to solve.
Well, even when you say animal biology, I recently read that it's mostly mice. It's not even, let's say, multiple types of animals, and so maybe mouse biology really is not very akin to human biology.
That's definitely true. Yeah, mice is really where they start. They'll ultimately try in other animals, but mouse is the easiest one to work with. But when you talk about mouse Alzheimer's, it's not too similar to what humans have, unfortunately.
All right. So these animal models have a tremendous failure rate. What's your premise? What are you working to do? Like, what kind of models are you creating?
So the models that we work with basically start with human stem cells. And these are reprogrammed adult cells that start with a skin biopsy. And some people who are smarter than I figured out how to take a skin cell, turn it back into a stem cell, and then differentiate that into a nerve cell. So that's what we start with. And obviously, the biology there is human. And we take that and rather than just looking at, a single cell or a handful of cells in a petri dish, we actually take them and we engineer this into a three-dimensional environment. The point of that is that it directly mimics, the environment and the interactions that a cell would experience in any of our bodies. At its base, that's really what it is. And we have two different platforms that we work with. We have a nerve on a chip, which is more indicative of cells or nerves that are outside of your spinal cord and brain. And then we have a mini brain that is actually mimics the cells that are, as you could guess by the name, actually in your brain. And I'm happy to go into more details about how that works, but it depends on how technical you'd like me to get.
Well, nerve on a chip just sounds to me like it's a lot easier than brain on a chip because the brain has so many functions. I don't know how you'd approximate all those functions on any kind of chip, but maybe start with a nerve on a chip. Are you trying to emulate all the functions of a nerve or just some of them? And, what does it look like?
Yeah, to your point of the brain is, more difficult to recapitulate or recreate. That's 100% true. You have so many cell types, so much complex organization in the brain. And even with the nerve, it's more complex and there are more types of cells involved than I think most people would realize. At some level, it boils down to making the best available model. Is it going to be exactly a human brain? Not quite. But it's close enough that it's better than animals. And so I will start with the nerve on a chip. And I think it's pretty easy to imagine, at least for me, although I've been working at this for over a decade, so I'm a little biased. Essentially, what we do by engineering this, as I mentioned, into this three-dimensional environment, we control the way these cells organize, and we control the way that this nerve grows, so that what we end up with at the end of kind of our development of the nerve on a chip is you have what looks exactly like nerves emanating or coming out of the spinal cord and going all the way to the tip of your finger. And just like if you touch something hot, that electrical signal goes all the way from the tip of your finger to your spinal cord. And that's exactly the data we are able to gather, although, in a lab rather than in an animal or in a human, like they would if they went to a neurologist. And what happens is if you have some kind of trouble with this nerve, then what you see is that signal slows down, going from the tip of your finger, again, to the spinal cord, and it also gets weaker. So ultimately, there comes a point where that signal might disappear altogether, or you lose sensation enough that it's extremely problematic. And the other component of that we have that adds a level of complexity that's quite unique is we have interaction with a cell called a Schwann cell. And what this Schwann cell does essentially is creates myelination. So just like with an electrical cord that you see, running above your street, there's a rubber wrapping around that, and that allows that signal to conduct from one point to another. You interrupted that the rubber wrapping, then that signal would basically die right there. So that's another level of complexity. We engineer that, and there's direct implications with that myelin in a number of diseases. So that's
in the human body. If I touch a hot stove, is there one gigantic nerve that goes all the way from my finger to my brain, or does it go from nerve to nerve to nerve and go through myriad pathways?
It does go through myriad pathways. That's a good point. At least the part of it, the pathway that is from the tip of your finger to your spinal cord. So that's sort of the first order of that pathway. That is one. It's basically unidirectional. Granted, it involves tens of thousands of nerves, which our components also are tens of thousands. Yeah, there's quite a few wrapped up, and if you think about your finger spread across.
I'm not talking about in serial though, like in parallel. Sure, I'm sure you don't just have one nerve in your finger, but in parallel, I'm sure there's thousands of them. But what about in serial? Like literally, if you just look at one nerve that experiences the heat, how many? What does it go through to get to the spinal cord? Like how many different cells does it go through?
Gotcha. Yeah, I was thinking in parallel, which is really what ours captures. Ours captures that first, basically from the tip of the finger towards what they call a dorsal root ganglia, right outside of your spine. From there, it does extend up into sort of the first order synapse into what's called the dorsal horn. And that's the part of the spinal cord that sends that the rest of the way up into your brain. Now, once it gets into your brain, things get extremely complicated. And there, I don't know that anyone exactly knows how many cells that might go through, but, in there you are talking about thousands of them, in any given, cubic whatever. And so once it gets to that point, it certainly gets diffused much more so. But the first two orders of that really are relatively simple. Tip of your finger to the dorsal root ganglia to the dorsal horn and then up.
So so you're looking to emulate what a real nerve would be like, but okay, so let's say you do that and you're testing it and it does seem to act similarly. Then what are you trying to test? Medications that can restore function if you damage that model you've made, or like what specifically are you trying to do once your model is working and it's somewhat similar to what actually goes?
Sure. There are two parts to what we do, and it depends kind of on where we are helping a pharmaceutical company. So one of them is starting from kind of the opposite of, what we're talking about here with recovery. It's actually looking at toxicity. So a lot of drugs and chemotherapeutic drugs in particular have, off-target side effects. They have unintended damage to a number of areas of the body, but the nervous system and the peripheral nervous system in particular is the main reason that a clinician actually will either dial back or cease chemotherapy altogether. So it's very, very problematic. And what that induces is what's called neuropathy. So what you see there, again, is that signal from the tip of your finger to your spinal cord. That is what gets interrupted. And so we help pharmaceutical companies. For instance, we're working with a company that is developing a next generation chemotherapy drug. They already have one on the market, and now they're kind of bringing out their, like I said, the next generation. And we're helping them by screening about 10 of their drugs that they have developed that they feel very confident in, that they want to take to humans, but they don't fully trust the animal models. And so we're able to put that into our system and get that conduction velocity, that tip of your finger to your spinal cord, and that lets them really predict what's going to happen in humans. And from that, we've helped them prioritize, a handful of those 10, kind of narrow that down to the ones that they think are going to be the safest. And then, they make the decision of balancing how effective is it with how safe is it. And ultimately, they'll be moving one of those drugs into clinical trials, with us being a part of the process, which really is our mission. And that's what drives us, is to enable these advancements in human neuroscience. And so the flip side of the safety, I was just going to say, the flip side of the safety, really is looking at protection or regeneration. So, if you know that one of these toxic drugs is going to go into your body, there's the idea that you can administer a drug at the same time to protect it. A number of companies are working on that. And then if you already have the damage, let's say whether it's from neuropathy, which can be from diabetic neuropathy, it can be age-related neuropathy, if you can introduce a drug that will actually help reverse that damage, that's where you really get to reach a much broader audience. And a lot of people, my grandfather suffered from neuropathy. A lot of people need help. It can be debilitating.
Yeah, no, I've experienced a little bit of it myself, but do you feel like you understand the mechanisms of at least one kind of neuropathy better than the scientific literature understands it, or does science understand it pretty well?
Neuropathies actually are pretty well understood. And, as far as mechanism goes, of course, with our model, it's not a fully intact human. There's not the entire system's biology. But the mechanisms that we're able to parse out from this really are, okay, are the nerves themselves dying back at the end? So, are the nerves at the tip of your finger dying themselves? Or at your spinal cord, are the nerve cells, the neurons, are those the one dying? Those are two completely different mechanisms, and depending on the type of neuropathy or the type of side effect, they're affected differentially. So we are able to parse that out. And then I mentioned that myelin before, that conductive wrapping. That's another area that's affected differentially depending on the mechanism. So we're not necessarily able to look at every single mechanism that may influence it, but the main drivers of either that damage or that toxicity, we are able to parse out and help these pharma companies understand, are they reversing it? Are they accidentally hitting some of these mechanisms, and help them redesign their drugs so that it works better?
Well, I know some of this is proprietary, but what can you say about the mechanism? Like, what has your model shown are the true mechanisms of X number of kinds of neuropathy? Choose one.
Yeah. So it's interesting, at the heart of this, it's science, obviously. And the way we sell this to pharmaceutical companies is they're scientists. So we have been supported by a number of federal grants, primarily the National Institute of Health. And what that allowed us to do is run a bunch of chemotherapy drugs that had been on the market for a significant amount of time. And because there's data from this testing, what it looked like in the lab to what it looked like in animals to ultimately how it performed in humans, and what we've seen tracks very closely with what was seen clinically. And what's interesting there is there's actually a few drugs that targeted the myelin, which is very unique to what we have, that did not get picked up in some of the laboratory tests, in these in vitro tests, because they didn't have the right interactions of the cells that produce myelin, whereas in ours, we do have that component. And we were able to see that would have happened before any other laboratory test would have seen. And now that same drug, when it went into animals, they did see some instances of this neuropathy or this neurotoxicity, but they thought that it was minor enough that they went into humans anyway. And then once it got into clinical trials, they saw up to 40% of patients got affected by this neuropathy, nowhere near what they saw in animals. And this was a while ago, and it was before there was many other options in chemotherapy, so they did let that go, and so it is actually being prescribed in the clinic, but clinicians are really not using this drug anymore because of those side effects. So if a pharma company had used us, we would have seen that neuropathy, we would have seen that mechanism of the myelination, and presumably could have either helped them stop that drug from moving forward, or helped them kind of reformulate it so that it did not have those side effects.
So how do you move forward? How do you become the standard? How do you get FDA process where the FDA says, use the animal model, but you also have to use AxoSim's model as well for drugs that affect nerves and drugs that affect the brain. They're required now because we've seen that they improve the likelihood that a drug will actually work. How do you get ensconced in that pathway?
Yeah, and I think you made an important distinction there, in saying that can we use these in conjunction and in parallel with animals? As much as myself and other companies like me and even pharma acknowledges, animals aren't good predictors, but at the same time, they are used now, they are well understood, and they do work sometimes. And frankly, the FDA is conservative, so we're not going to replace them immediately. But being able to supplement them and reduce that animal testing while increasing the efficacy is what we're looking at. And I'll say we've had conversations with the FDA. They're really excited about technology like ours, and they're excited to be able to use it, but they're very conservative. And, I would say rightly so. We don't want the FDA to kind of just willy-nilly let new technologies in and without validating them. So the burden is going to be more on ourselves and the pharmaceutical partners that we work with. So the FDA said, hey, we're tracking this, we're interested in it, but we're not going to be the first ones to just put our rubber stamp of approval on it. Instead, there's another company that's doing heart on a chip. We know that they have worked with a pharmaceutical company to generate a set of this data around safety, so around cardiotoxicity. And we've heard anecdotally that this data looked so strong that this pharmaceutical company actually has brought it to the FDA and said, hey, here's what we saw in this heart on a chip. Here's what we see in animals. And, here's what we might have seen, in some other laboratory-based testing. And all indications are that the FDA is actually going to use that data as part of their decision-making process. So that's step number one. And now if it can happen in heart on a chip, I think that lowers the barrier for somebody like us with nerve on a chip. So, rather than the FDA, like I said, just rubber stamping this, it's going to come from the other direction. It's going to come from the push of pharma saying, look, this technology is better. This technology saves us time, and this technology saves us money. Let's work with us, FDA, and tell us exactly what you need to see.
Well, since there's no one being hurt, no animals, no people, nothing, and can this be used anyway? You need the FDA's permission to use this. Can you just use it and submit the data, whether the FDA takes it into account or not? Is it a might as well type thing, or do you need permission to even do this kind of stuff?
So we don't need permission to, work with pharmaceutical companies. We already have a number that we're working with. And the reason for that is we kind of help them make their decision-making process easier. So to go from, 50 drugs to 10, and then from 10 to the one that they want to take to the FDA. Now, the FDA's official stance is, you can put anything into a package you want. They have guidelines. They don't have regulations. But frankly, these pharmaceutical companies don't want to put data in there until they've talked to the FDA about it, just because they don't necessarily know how the FDA is going to approach that. So they're also conservative about what they put into their package, until they've had those conversations where the FDA says, this data looks good. I think this data will make us stronger.
Well, why not a two-pronged approach? Why don't you approach the FDA and get a list of, 20 failed nerve-acting drugs, and you say, hey, FDA, we'd like to run these through our nerve on a chip thing and see how it correlates with what was actually found once these got to that last step where they failed. Maybe that's another angle that would get faster adoption of something like that.
It's definitely another angle, and it certainly is one that we're working on. Like I said, we have talked to the FDA. We continue to talk to the FDA, and the FDA technically has put out, a roadmap to adoption for technology like ours. Now, unfortunately, what the FDA has not cohesively either figured out for themselves or told us is number one, how many drugs do they want to see to accept this, or really even what drugs are they that they want us to test? So we're building up that data set, we're showing it to them, but they have no criteria themselves to say, with us approaching them, okay, that's enough drugs that we test it. So it comes down to a bureaucracy and a conservative nature of the FDA, where they're not going to let us work directly with them, but put together a better package. So if that makes sense, it's something we want to do with them, but it's something that we want to do with them and we're trying to work on, but it's unfortunately not that stuff.
Yeah, it doesn't make sense. If the FDA wants, if they want to be able to do their job better and get better outcomes, they should be able to develop some goals and then share them with them. Say, hey, we want this, whether it's unreasonable or not. But how are you supposed to please them if they don't even know what they want? But I guess that's unanswerable, unfortunately.
You ask an excellent question and make a great point, and I wish there was an easier answer.
So what's the ideal? Your nerve on a chip stuff, for instance, works. Is it better to do it before animal testing, in parallel, after animal testing? Like, what would be the ideal that you would think? How should a pharmaceutical company use it ideally?
So what I can answer that by saying, how is it being used now, and how do we want it to be used? What we're finding right now is it is being used in parallel with, and in some cases after animals, and both of those situations are because these pharma companies want to get comfortable with it. So this is new technology, and similar to the FDA, the pharma companies are quite conservative, surprisingly so, and they want this technology, but they want to vet it. And we can go up to them with a set of our own data to say, hey, we tested 25 drugs. That's enough to get them interested, but they still want to see it for themselves. So they'll typically test us alongside some of their drugs that they know how they've acted, or know how they expect them to act, as well as some drugs that they don't know. Of course, along with controls as well. And that has allowed them to build a comfort level that we are now, at least with one of our customers, working before animals. So helping them pare those drugs down so that they have a better confidence to then move to animals, which of course saves them time and money. But they're not quite ready to replace the animals, certainly. And ultimately, that is where we want to go, though. If a pharma company's got 100 drugs, and right now they're really only able to afford to or are comfortable with testing, let's say, 20 of those in animals, if we can operate as effectively as we think we can, and as we've seen that we can, we can let that pharmaceutical company test all 50 of those drugs instead of really having to automatically pare it down to 20, and then say, oh, now we feel really confident in this 10, and we'll take that into animals. And presumably that will translate to better success. That's where we want to go.
Well, what about their closet full of failures? Do drug companies just give up? Or, that's a whole opportunity in itself. All right, give me all your failures, and we'll run our nerve on a chip stuff. And then you can run experiments to make modifications to the drugs that failed, and maybe that'll be a much quicker path to get you a success out of the whole list of failures. Will they do that with you more likely than other ones that are earlier in the process that they haven't failed on?
So we are actually doing that with one customer, and it's a really exciting project. So this particular pharmaceutical company had basically, they had a program, a number of different analogs of a drug that it was an oncology drug, that they were taking into the clinic. And unfortunately, what they saw was conflicting data in a number of different animal models. They did see these neurotoxic side effects in one or two animals. They didn't in one or two other animals. And they just never got comfortable with moving the drug forward, even though they really, really thought this thing was going to be effective. We are working with them now and are actually testing some of those compounds, and we're getting them comfortable enough that they finally do now have a tool to have the comfort level to move this thing forward. And so we're potentially unlocking an entire portfolio of drugs for them that they had completely shelved. So that's absolutely a level and an area we want to work on, but that requires a good level of trust and confidence in what we're doing.
Well, what about for you internally? You have your nerve on a chip and your brain on a chip. Again, me not knowing anything, I would think the brain on a chip would be much, orders of magnitude more difficult, but maybe there's a trade-off there. Maybe it's more exciting to the pharmaceutical companies, or there's more money there. I mean, what have you seen? Which of those two systems do you think is going to be the big winner for you, and is going to get faster adoption and will make more of an impact?
So ultimately, the mini brain technology probably has the broader reach. The nerve on a chip is what we developed early on. We licensed that technology from Tulane University, and that was actually based on my PhD research. So I've been working with this technology for 12 years now, which is exciting. The mini brains we just licensed from Johns Hopkins, from the laboratory of a researcher named Thomas Hartung. So it's we're really just getting this off the ground in our lab, but from the proof of concept that he's had in his lab, that he's already proven that this technology has relevance to multiple sclerosis. It has applicability in glioblastoma, in Alzheimer's, and Parkinson's. And so, once you start getting into those diseases, there's such a need, and there's such devastating diseases, and frankly, pharmaceutical companies are working on areas of those drugs. You can look at Alzheimer's in particular. The number of failures is staggering in Alzheimer's.
Oh yeah.
Unfortunately.
Oh, yeah.
And it's because they haven't had the right tool. And so this mini brain potentially unlocks that tool. So it's kind of a trade-off, right? It's the more complex system, so it's a little bit more difficult to tackle, but the upside is huge.
Yeah, I mean, within mini brains, what part of the brain? The amygdala, the hypothalamus, the cortex, 2 parts of it, 3 parts of it. I mean, it seems like there's so many different parts to model and so much different functionality. How do you even approach that without, like, I don't know, just being lost in the complexity of it?
I guess the short answer is you could easily get lost in the complexity of it. And so to not get lost in the complexity of it, you have to kind of go back to what's the best tool available. Starting out with a very defined, well-characterized model and building from there. So the mini brain right now really is not a highly specific part of the brain. A lot of people call them cerebral organoids. I would say they're really more applicable, for the cortex, cortical spheroids. But again, they're a little bit broad, and that is good because it gives you a little bit of reach and it allows it to be characterized. But where we go from there is becoming more specific with the areas of a brain. So, from the academic side, people have figured out protocols to differentiate those so that they are like certain regions of the brain in particular. And that's definitely an area we want to go, but taking that, validating that, characterizing that so that a pharma company is ready to use it takes a long time. Now, another way that we are building in complexity with these mini brains, so right now they have 3 different cells, what I would argue are the 3 most important, broadly speaking, cell types in the brain. They have neurons, so they have glutamatergic neurons, GABAergic neurons, they have nicotinic, and then they also have astrocytes, and then they have oligodendrocytes. And those oligodendrocytes are what makes it the most unique because no other commercially available model has those. And then that's also what myelinates the brain, which has implications in diseases we know for sure from multiple sclerosis. They're starting to identify that also has implications in Alzheimer's, which is interesting. And so, we start with that as our basal model. What we find is we can then introduce other cells into the system to increase the complexity. So one area that's really hot right now and that people think are implicated in way more diseases than originally thought is microglia. So if you're looking at inflammation, neuroinflammation, and how that plays into a lot of diseases, we can actually add these microglia into our system. And immediately there, you have more complexity, you have more applicability. Another thing, I mentioned glioblastoma before, we can take actual patient biopsies of glioblastoma, introduce those into our mini brains, and we see that they do grow and manifest in the mini brains. And then when treated with some of the first line therapies, you actually see that they shrink and they respond. So, the first step really is just being able to add in selectively what cells do we want, what cell types are we interested in. And then, I think as the field and the technology grows, bringing in some of that where we can differentiate this into specific regions of the brain and look at diseases that might preferentially affect those.
I mean, since you can't do everything, especially in the brain area, are you sub-licensing? Like, you're getting good at this organoid on a chip type stuff, but have you considered sub-licensing? Like, the brain, there's no way we can do these 50 different configurations. So have you talked to other companies that are trying to do organs on a chip, and maybe you do the, these 5 flavors of a brain on a chip and you sublicense your technology to other people that do these other 35 flavors. So you get more coverage and, maybe you get a revenue stream from that. And that way you don't have to do all the work.
So the way we've approached that, I would say, is really kind of the opposite. A couple of reasons for that. The industry is still emerging, let's say, and the majority of the players, we do all know each other, whether it's other companies working on neuroscience, or it's other organs on a chip. And, as this company's emerging, this technologies are emerging, I think everybody's still trying to figure out how does everybody fit in there. So while we know what each other are doing, we really haven't explored those those approaches, the sub-licensing, because I think there's so much potential to develop these internally before we start giving out parts of it. We've actually taken the opposite approach where we're scouring academia and we're scouring, other companies who might be maybe a little bit younger or emerging but floundering and licensing their technology. So our mission is to be the company that every single pharmaceutical company looks to if they're developing anything in the neurology or anything that has neurological safety implications. So we do want to build that company, recognizing we're not going to be able to do all that internal R&D. So we want to be the ones, and we already went to Hopkins and licensed this mini brain technology. Along with that licensing, we actually acquired a company that the inventor, Thomas Hartung, had already spun out, and that company really laid the commercial groundwork, but he saw, the potential in our company to be the ones that really hits the home run with it and takes it to the market. So that's the approach that we prefer to take because we want to be the industry leaders.
Well, like in terms of the nerve on a chip, what about nerve regeneration? That's a whole other area. I don't know if, I just wonder, how do you have time to do all these things? So one company, it doesn't seem possible unless you have unbelievable resources to do it all.
Yeah, the answer, unfortunately, is you can't do all of these things. We kind of learned that early on when we did stretch ourselves a bit too thin. So it's a push and a pull. And myself, I'm a scientist at heart, so I do want to do everything. I do want to chase the interesting questions. But you have to pull back and say, no, what do our customers want? Not what are all the million capabilities of this. And so interesting that you talk about nerve regeneration. When I was working on this as a PhD project, we actually were building a model for spinal cord injury and how you can influence spinal cord regeneration. But the fact is, if you're talking about that as a commercial endeavor, you're talking about a clinical pathway that is extremely expensive, extremely time consuming, and frankly is a bit crowded. So that's why we realized that technology really fit better into an application that was untapped and had a big need. And that was the drug screening. And so that's really where we've hit the market. Now, are we still cognizant that there could be other areas like nerve regeneration where this could apply? Sure. But for now, we're trying to stay laser focused.
Gotcha. Okay. Well, very good. So what do you think is possible in the near term and in the further future? And you define it. Near term could be a year or 3 years or 5 years, far term further than that. So, what are some general things that you would be so excited if they happened? What do you think is possible?
So ultimately, let's start with long term. And I think there's two things that we get excited about. The first one is really to be able to point to a drug that comes out that truly is a cure of let's name the horrible neurodegenerative disease. Let's start with Alzheimer's. Our mission really is to be able to point to a drug that goes through clinical trials that is ultimately successful and cures this debilitating disease. And once we can point to that, we have accomplished one of our biggest goals. There's still many, many other diseases to tackle, but you're talking about a clinical pathway that's a decade plus. So that certainly is a long-term goal. Now, if you start dialing this back to shorter-term goals, personalized medicine is another area that we think kind of in the medium term that we could have a serious impact. If you look at a number of neurodegenerative diseases, they're really a family of diseases. When you talk about genetic disorders like ALS, it's not just one gene, and it's not so easy to identify which genes are at play. So, if we can go to a patient, we can get, a skin biopsy, ultimately turn that into nerve cells, and then allow a clinician to screen, a number of drugs that they might have tried on the patient and already figure out, in a matter of months rather than years, this treatment is going to be the best one to go into our patient. We don't necessarily know why genetically, but it looks that way, and then put that into a human, that's certainly something that's doable, and it's something that's happening in other areas, cancer being a big one of them. There's a company called Biotech who actually has a clinical trial going on right now where they are influencing clinician decisions on therapeutic treatments. And then, you start dialing back again to shorter term, let's say over the next 1 to 3 years, really what we're going to call success is broadening our platform so that we're reaching more of the market and having a larger potential. So right now, if you look at the nerve on a chip, neuropathies really are the main area that we're focusing on. There are some rare diseases like Marie-Charcot-Tooth disease, Guillain-Barré, ALS has some applications with our nerve on a chip. So we want to be able to address all of those, be a better company, and be able to have more impact. Then if you look at the mini brain, I already talked about some of the models, and we want to have pharmaceutical companies using our MS mini brain, our ALS mini brain, our Alzheimer's mini brain. And that gets us on the way to personalized medicine and certainly on the way to getting approved drugs out there.
Okay. Well, excellent. So what's the best way for interested parties to get in touch, to see what you're working on, to interact with you somehow? Website? What do you suggest?
Yeah, no, website, definitely. We have some information on there. You can learn a little bit more about what our models actually look like. And when I describe a nerve on a chip or a mini brain over the, abstractly, it's kind of hard to see, but we've got some really nice videos on our website. Certainly have a way to get in contact with us through the website. But then, we are also active, our Twitter account, we're always talking about what conferences are we at. We really try to bring important papers that are coming into the industry, whether they're ours or not, out there and really try to, raise awareness for the organ on a chip industry and then, progress that's being made with a lot of these other diseases.
Okay. Well, that's great. Well, I appreciate you coming on the podcast. It's been good talking to you. Thanks, Lowry.
Absolutely. Thank you very much.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.